morning, everyone, and thanks for being here. So it's my great pleasure to introduce Professor Koen Van Leemput. For those of you that doesn't know him, he has a background on electrical engineering, where he completed his PhD in the Catholic University, whatever, in Leuven, Belgium, in 2001. And for me, what it was more impressive at the time that I was starting my PhD, it was that he had actually not one, but two, but three papers in TMI of the same year, that I don't know if many people can have them. But the most interesting one for me was the one that you published the year after on the partial volume. Hello, Pina. Then he moved to the Helsinki University Hospital. Until 2007, he was as well starting from there at the MGH, the Martinez Center for Biomedical Imaging and Harvard Medical School. Then he moved to the Technical University of Denmark till 2023, where he has now a position at the Aalto University. He's actually part of the editorial board of the most prestigious journals in the field, like TMI or Medical Image Analysis. And he is as well program committee at several conferences of MICAI. Maybe you don't know, but he was actually the leading researcher launching scientific challenges, especially BRAT, that has more than 7,500 citations. And personally, I had the pleasure to closely work with you for a training network, TRAVIT, that will repeat the experience again, where he has been leading these two training networks, Marie Curie, that we will start now. And I'm very happy to continue working with you and that you're here. Please start with the talk.
Yes, thanks really for the nice introduction and for hosting me for several weeks now in your lab already. So my talk is about sort of two things that are maybe a little bit unusual in medical image analysis. One is generative models, and the other one is try to push generative models to the extreme of simplicity while still trying to make sure they can work. And I'll try to motivate this choice a little bit. So if you look at what's happening now, I think by far the most methods are descriptive methods, which means they're essentially example-based learning, which in reality really means it's not really much learning going on. It's sort of more multi-see, multi-do. So it's a bit like, solving exam questions in math without actually understanding the problems, but you have a secret stash of previous solutions. And then if a math question comes up, you look what the best matching answer is, and you sort of replicate what was given before. And so that has a number of implications. One is about generalizability, as you can imagine. If someone suddenly asks a very different type of question that you haven't seen before, now you're in trouble, right? You don't know how to solve this. So that's in terms of generalizability. I think everybody knows that discriminative methods struggle with that. Also interpretability. If suddenly someone asks now, well, can you explain why you gave this answer? And essentially you don't know. You just said, well, this question was answered like this, and this is similar, so I'll give a similar answer. And I'll give examples of how that works out in practice later. And then finally, in terms of uncertainty, it's probably going to be easy to say, well, this image data is actually different from the examples that I've seen before. It's a different thing to actually quantify how much information there really is, or lack of information there is in the images. And so, I hope I'll be able to convince you generative models, they don't have many of these problems. So generative models kind of turn the whole thing upside down. So instead of going straight from images to extracting relevant information, we try to say, well, if I know the reality, I know the age of this person, or I know the disease, what sort of images do I expect to see? And when you have that information, you can try to invert the model to find the best matching explanation. So the other thing I sort of stressed is that I want to look at simple models. And I think many people will agree with me that if you look at what's been happening in the field, it's always bigger models, more compute, bigger data sets. So as Jonas pointed out recently, at Mika there was a challenge. They had over 100,000 brain scans pre-training. There's a lot of things going on. And I sort of want to now sort of take that extreme and push it all the way to the other end. And sort of try to find the simplest models that we can use that still work, and actually it works surprisingly well. And there are several motivations for doing that. One is, I think, if you take a model and you remove all the bells and whistles until you're only left with something that if you remove one more thing, the model starts failing, then you need to learn something. Right? And the other thing is that very simple models can actually work very well. And I show you an example of the bias variance sort of trade-off. So if you have a very complex model, on average you're correct, but you never sort of hit target. Every time you train on a limited set of training data, you'll get completely different answers. The other extreme is what I showed there on the left, which are very simple models that are biased. One example would be the naive Bayesian classifier. But if you don't have the training data, it actually works surprisingly well. And then finally, that's kind of more my personal motivation. If not, everybody has to work on the same thing. So for those people who are not so familiar with this kind of very simple models, so I think we've been a bit spoiled in recent years. All you need to do now is come up with a cost function and say, press the button, something behind the scenes optimize it in very generic terms with gradient descent. Once you go to these very simple models, you can actually squeeze out more juice from understanding what the problem is. And an example I showed there is optimization, where essentially you have an objective function shown in the blue, and you want to optimize for the parameters theta. And one very common trick, there's a bag of tricks you can use to create an upper bound, shown in whatever color that is, green or orange. Sorry, I'm colorblind. And so the trick is that this works or this doesn't work. At the current value of your parameters, the upper bound touches the objective function. And then if you can then optimize the upper bound, you automatically have increased or improved the real objective function. And so the trick is to choose upper bounds that are easy to optimize. Often we're given optimizations given, or the optimal values given to us forms. So we've taken a difficult problem and turned it into break it down into many small, very trivially problems. And one example is Ian. I'll use that later on in the talk with several completely different areas. And I just want to give the gist for, I guess most people know what Ian is, but just as an example. So it's one of the tricks that you can use to generate this sort of upper bounds. And it's a trick that comes into play when you have latent variables. So, and I show there on top how that works. So imagine that you have a binary image, and the label in every pixel is just a flip of a coin. So it's heads, it's foreground, it's tails, it's background. And then you associate for the foreground and the background Gaussian intensity distributions. And now the problem is, if someone gives you the intensities only and tries to say, well, what was the median variance of the foreground for the foreground class? And so the trick is that if you knew the latent variables, if you knew the labeling, if you knew what the foreground was, then it's easier. You just compute the median variance. Of course, you don't know that, but then you actually you can compute with this EM algorithm, this upper bound, and optimize that, and then you'll get sort of closed form updates with a guarantee to never step in the wrong direction. Beautiful algorithm if you think about it, and very easy to do that. Okay, so I'll be talking about three different things, completely sort of different topics, where I sort of highlight the power of these generative models. One is about brain segmentation, the other one prediction, and then finally about image distribution. Let's start with brain segmentation. Here I'm going to really focus on how generalizable itself. So imagine you set yourself the task of developing a computer program that can actually automatically segment all the brain scans that are required in the hospital. And then you think about all the sort of sources of variability that you can take into account. And the first one, on the top left, that's the obvious one, that's what everybody's nowadays very concerned about, is about, if you have different sequences of different scanners, if all your training data in a distributed method comes from P1 weighted data, and then so on, as the two-weighted scanner, big problem. But there is other things that cause variability in the data. One, which is actually on the top right, which many people don't talk about nowadays, is just scanner different resolutions. And many challenges they pretend that doesn't exist. All the data is 1 millimeter nowadays. That's of course not true. Then you have different pathologies. Maybe you see someone with a brain injury, someone with a stroke. Some hospitals they don't require just one data set, but actually different contrast there. You don't know in advance what contrast they're going to require. There's longitudinal data. Some people you've seen only once, other people you've seen 5 times before. And ideally, you try to sort of use all the data available, or use some other modalities, maybe diffusion imaging, CPU, and so, if you want to do this with distributed methods, it's really a big problem for yourself. And so, what I'm going to try to convince you is that using generative models is probably the more natural approach to solve this problem. I'll give you an example of a very simple generative model that actually is still very useful in practice. It is a classical sort of approach, so there's nothing fancy about this. So you start with a probabilistic atlas, which actually is some sort of average anatomy, sort of locations of probability of 40 different structures. And then the model is that you generate randomly some mark fields, some atlas information, and then you pick in every pixel in a correct location the label from the discrete distribution, essentially you make a segmentation. And then in order to go from segmentations to real intensities, you'll associate from distributions, typically mixture models to each structure. And there's some artifacts that you typically have in the MRI, bias fields, and you add everything together, and you can sort of see the simulated image. Doesn't look very realistic, but it actually does. And so you can do it. This looks like a P1 weighted image from this week. Give it the same for image. So how can we use this type of models to then go the inverse direction? Given a brain scan, what is the underlying segmentation? One that has got a lot of attention recently is StimSec, I think many people heard about, developed by colleagues of mine in Boston. So what you do is essentially you use this model to generate lots of pairs of real or simulated segmentations and simulated corresponding intensities. And you just keep generating lots of different data, right? So it's how it works. And if you train this thing during a week, then you get a network that knows how to convert this process. And you can get segmentations pretty quickly. Okay, so let's see what sort of area we've now covered with this sort of technique. So we've certainly covered the thing about P1 versus P2 versus whatever contrast you might have. The seven models. The resolutions, it's a little bit yes and no. So they actually have a very cool trick where their forward model is a little bit more complex than what I showed. They actually have a blurring step. So if you have thick slices, then it can actually virtually upsample the data. It's segmentations at high resolution. On the other hand, what they do is resample everything to 1 millimeter isotopically. It's the first thing they do. So whatever the resolution of the data, you always get segmentations at 1 millimeter. Maybe here that's fine. If you have a 0.7 isotopic scan, you're not so happy with the segmentations. And so all the other stuff it's not covered, even if you already trained 1 week. And so the problem is that there's no compositionality, which means that every case that you would like to cover at training time, you need to sort of think of, remember that, okay, this case we need to cover as well. Maybe you want to segment multi-contrast data from the demonstrations as well. Maybe you want to have longitudinal data, but every time you add one of these dimensions, the training time will grow exponentially. And so obviously that's not realistic. So what you end up doing is for each new target, it's a brain tumors stroke training. And I think that's far from ideal. So here's another solution. I think it's better. I don't think many people are convinced of that, except for maybe myself and theoretical. And so of course there's another option which just do the inversion manually. So you use Bayes' rule, try to approximate what the underlying segmentation must be if you see a brain scan. And so the key to the method is that you can adjust the model as needed on the spot. So instead of saying, oh, you give me some data that's kind of a corner case I've never seen before, oh, damn, at training time we didn't think of including that. We just say, oh, no problem. Yes. Longitudinal data with 7 time points and with 3 contrasts that I've never seen before. That's not a problem. Here's the model, and we try to do it. So there are several ways of building it in practice. So we have software, it's doing research, it's Ellipse, which is one of the sort of major packages to simulate electrical fields by stimulating the brain from the outside. So for shape, we have different meshes, just a general purpose optimizer. And for all the other parameters, maybe sort of closed form EM sort of nice. These are all algorithms that do a very low gauge. And you can actually get segmentation in a few minutes to CPU time. So, assuming if we would have the energy and the resources to do the GPU. And so now you can do things that you couldn't dream of using discriminative methods, or at least it would be very difficult to do. So we have training data that was acquired in Boston in the 90s, manually generated by someone who spent every year of her life doing just that. And we did models that actually now segment multi-contrast data with different resolution from a completely different machine. We just got scanner that we didn't use back then. And actually get very good segmentation. And here's where this compositionality comes in. So let's say we want to take that and say, well, okay, I have now a patient, he's in MS patient, multiple sclerosis, this person has these lesions. Instead of starting from scratch, we just say, well, let's pre-build a model of where lesions occur, and what sort of their shape looks like typically, and then you have models of different disorders, for brain tumors. And when an image comes in, you just take from the brick shelf, the model, you stick it in, and use all the machinery that's already available to get both the lesions and all the other structures. So what you can do then is get some data from France. This is from some national cohort study, I think. So here they happen to have 5 different contrasts. They're interested in the lesions, and all the neuroanatomical structures. Then in Denmark, they only have T1, T2, and flare, with different scanner obviously. And this kind of works out of the box. There's no recreating. It's just very simple logic. Here's an example of the same thing using brain tumors. So once again, you don't start from scratch. You actually have a model of brain tumors look like, and sort of combine it on the spot to sort of invert the model. And on the right, I just show the application where we try to predict long from short survivors with the glioblastoma by just looking at the shape of the surroundings. And then the last example I'll give, sort of the power of sort of generative on the spot sort of models, is in longitudinal imaging. So there's on the left scan Alzheimer's patients, an Alzheimer's patient that we've seen several times, and on the right, well, there's this patient. Of course, what you could do to simulate this data is take one time point at a time, push it through whatever method you have, and then see how the volumes change over time. There's a better way to actually model the whole thing in one go. And you'll notice that if you look at the time places for the Alzheimer's patient, so the baseline scan, then more than 2 years later, had another scan, and had another one. So the time difference is not together. And this person had 4 time points, the MS patient had 3 time points, lovely spectral data, and we actually just. And I will not explain everything that's on this slide. Essentially, if you look at, for instance, you say slope of change, rate of change over the years between normal aging and Alzheimer's. If you model things, longitudinally, you actually get much more significant. Okay, so let me switch gears a little bit and talk about the second topic, which is subject level prediction. So here the focus is on something completely else, interpretability. If I show you a scan, and I try to ask the audience here to estimate how old a person is, it's one person we get. You don't have to be wrong.
30 years old.
30? But that's the always thing that people are young. So this is kind of the real age of this person is 47 years old, and with the computer you get some 45. That's kind of a typical error that you can get nowadays. So there's something strange going on though. So first of all, you don't need very fancy methods to do this. Even just a linear method will work just fine. And I'll show a result to prove it a little bit later. But if you just look at what's happening in the linear method, if you train on 300 subjects, that's the image on the lower left, there's a color map that shows that essentially the weight that the method is giving to specific voxels to generate the age. So if in the blue areas there is something lighting up, it means that if you have dark intensities, your age is going to be predicted to be older. And then the weird thing is, first of all, that's kind of not what a human expert would sort of look at to predict the age. And the second thing is that if you change the number of subjects, different areas become important to make a prediction, which is kind of, you can do the same stuff with a nonlinear function. And so first of all, you have the same problem, and also there's no correspondence between what the linear method used and what the nonlinear method used. And so the solution here is sort of modeling things in the correct direction, basically. So you know that age causes brain changes and not the other way around. And that's kind of the root cause of this sort of bizarre sort of result you're getting. So if you build 4 models, sort of generative models in the correct direction, you'll see you don't have these bizarre problems. Actually, the method will actually say, well, I'm looking at CSF spaces. If they grow, then typically you're an older person. And this is actually a very well-known sort of thing. And just to give a toy example that I show from one of the papers shown there, imagine you have an image with 2 pixels, and only 1 pixel actually has some disease effect. So let's assume the color in blue is controls, and in red is a patient with some disease. So in the second pixel, so the second feature there is absolutely nothing happening. If someone gets the disease, the second pixel doesn't change. And what I also draw there is just a very simple model. There's some noise which models the individuality of people. Even among controls, not everybody looks identical, so that's modeled there. And so this direction 0, 1, it's kind of the stuff that's really going on. And I think many people will know that actually if you choose the optimal classifier for this, it's actually logistic regression classifier, a linear separating hyperplane. And actually to compute, you actually use a different vector. So I call that sort of interesting vector on the left, I call that WG for generative. And here that's, if you want to make predictions, you need to actually have another vector I call WD, which was for discrimination. And what you need to do is you need to take the generative vector multiplied by the inverse of the covariance matrix of the noise. And that's kind of giving you a prediction. So what you're doing is essentially, you're trying to find the real signal, so what's disease effect, while ignoring the noise. And so it kind of makes sense that you would look at the second pixel, because if you know the second pixel, you have measured the noise, so you can predict what the noise was in the real stuff that you're interested in removing. And so the interesting thing in this example is that even though the second component has no information of the disease, or it has information, it has no causal effect of the disease, it actually gets, if you do the math, so the discriminative vector has a higher weight for the second pixel. And so the difference is that on the left, it's about interpretable AI. So, what's going on? Why would you predict that this person has a disease? And on the right is what does the method do, which is something else than saying what the model, what the world is doing. Okay, so there's a bunch of things that people have tried to do to get around this. So in practice, you can't have these sort of simple models. You can't estimate huge covariance matrices. So what people typically do is this train a descriptive classifier and then ignore the problem to say, hey, look, he's looking at these sort of voxels, and therefore this must be affected by disease or something. And there's actually a very well-known trick called the Howfoot transform that many people in neuroimaging use, which is kind of inventing a generative weight that's kind of compatible with the discriminative one. I won't go into the details, but essentially, once you understand what it does, you would probably never use it. So the experiment we did is, if you look at the math, what's happening behind the scenes, but it's not obvious from the expression, is that you're essentially using predicted targets rather than the real targets to fit the generative model. And so one experiment we did is just predict age based on only 1/2 of the brain, and you can predict very well because it's kind of redundant the other side. And if you then generate sort of this compatible generative model, you'll see that it claims that actually you're using the right part of the brain as well, which obviously is not true. So we came up with a very simple solution. It was so simple that during peer review we were called crazy and naive and all those type of bizarre things. But it still works, so I think it's a valid sort of point that we made. So the problem is you have this noise covariance matrix in high dimensions. You can't estimate that, but there's well-known tricks. It's closely related to PCA. And it's a latent variable model. And for people who are familiar with radiational autoencoders, it's just the baby version, the linear version. And it's very simple, so it has a latent variable, so you can use EM to get beautiful closed form update equations. Very easy to optimize. And then if you apply this to biobank data to predict age, you'll see what the model really does. So it has this sort of generative effect that's shown in the top left, which is kind of what a human would look at to sort of make predictions. And then importantly, it also has sort of the major components of the noise. And so the first mode is just how bright is the image. And the second one is sort of, there is some bias, it's bias field corrected, it's pre-processed biobank, but it's still there. So it detects, okay, images differ because sometimes it's a bit more dark on the top and sometimes a bit more bright. And so it tries to make the age prediction, it tries to look at the top left figure, how big are the ventricles basically, and trying to ignore all the stuff that as a human you would also ignore. And as a result, mathematically, it uses this linear map that I show on the right. But that's now completely not interpretable. It's just a recipe of how to compute the result. It doesn't tell you anything about what's really going on inside the images. Just to show that it works, so we applied it to biobank data. X-axis on the left plot is the number of subjects on the logarithmic scale. And effectively, I mean, if you ask me, there's not really any difference between the methods. So with we compared 3 different methods: linear discriminative, nonlinear discriminative, essentially a neural net, feed forward neural net, and then a linear generative one. And they kind of perform the same, and it's really, you're playing really with this sort of bias variance trade-off. So our method is way too simple, it has a high bias, but then it's very stable. So in the end, it works just as well as a neural net that sort of starts doing crazy things if you don't have enough training data. And by the way, you know, before you say, oh, but UK Biobank is scanning 100,000 people, if you want to do anything else than predicting age or gender, if you want to look at diseases, you end up with small data sets. Like there's going to be like 300 MS patients, I think, expected once you scan 100,000 people, and something like 1,000 epilepsy patients. So it's a kind of dream that you get these huge databases for and predict lots of different diseases. That's not going to be the case. Okay, so then maybe you're asking, okay, you have linear discriminative, nonlinear discriminative, linear generative. What happened to the nonlinear generative? Well, this is what happens. We tried. So you have this, in the linear case, you have this beautiful EM algorithm, everything is given in closed form. Once you make it nonlinear, you still have an EM algorithm, but you have lots of problems, you make lots of approximations to get the upper bound. And then you need to do sampling in the E step, and then, you know, gradient descent to, yeah, lots of problems. It's way slower, and accuracy goes down. So just one example of where too much complexity can really kill it. Okay, so I'll quickly go through some extensions just to show why there's cool stuff that you can do with this. So this ongoing work. So what I show here is classification task between control and Alzheimer's based on 2 volumes, hippocampal volume and intrarenal thickness. And so now every patient is sort of a little trajectory over time. And so what you can now do is you can try to estimate the arrows of time. So what's the effect of time on controls? Obviously, your brain shrinks a little bit. For Alzheimer's, your brain shrinks, but faster, and you start at a different starting point. And then there are mixed effect models if you extend this. So essentially, you take the expected trajectory of someone, and you can move it as a translation somewhere randomly around. That's how you. And then there's the same. That's what I tried to show with the ellipsoids. And then there's the same, but I can't show it is, you know, you can also estimate that some people are just faster in developing with aging than others. And then if you then get a test subject, which is also just a little trajectory, you can try to exploit the temporal information to get higher accuracy in classifying. It works not as much as you would think. You know, if you know the history of someone, it doesn't help as much as I think I was expecting, at least. But here's another thing that's maybe more interesting. Here we have the same experiment, but we have the same data, but we just remove the labels. We just say, here's a bag with subjects. There's 2 classes in here, find them. And you actually can find it, right? And so in high dimensions, this is what it looks like. So this is cortical thickness changes over time in controls versus Alzheimer's disease, even without telling the algorithm which is which. You just give a bag of subjects and say, find some clusters in this. And the reason that this is easy to do is because we use an EM algorithm for the factor analysis. And it turns out you can use the same, you have additional latent variable, which is which class do I belong to. You just stick it in the same algorithm, it will find the sort of prototypes. Okay, so I have one more topic, not that many slides. So it's about something completely else again, registration. So in here, I'm talking about generalization. I'll talk a little bit about uncertainty as well. So imagine you want to write an algorithm, accepts 2 images and aligns them. And of course, if you have segmentations in your training data, everybody knows, I think now, that deep learning blows everyone else out of the water, because it's implicitly it can sort of learn to segment the images and then use segmentations to drive the registration. Of course, that's not really the settings that we're dealing with. Our task is give me two images. They might be from lungs or the brain or who knows what part of the body with whatever sequence or modality. Can you align them? So the promise of classical algorithms is that yes, we can do this. We have algorithms that are adaptive to contrast. They can accept different image resolutions. In practice, it doesn't work quite so well because there's lots of knobs you need to tune to sort of optimize each algorithm to a specific case. So you actually might still need some segmentations to improve some die score and some validation set. And so what we said, well, okay, let's try to simplify this and try to find something that still works, that's as simple as possible. And so we started from mutual information, which is kind of a very well-known algorithm. And especially there's a formulation of mutual information that formulates this as a generative model. And so the way it works, I took a CT image and I essentially say this CT image has generated the MR image. And just for illustration purposes, I quantized both images in 6 different levels. So there's only 6 different intensity levels in each image. And so the model works, the way the model works is that you have some sort of deformation field. In this case, it's pure rotation. For every pixel in the fixed image, I look where I land in the moving image in the CT scan. Look what the label there was, the label of the nearest neighbor. I get a label, and then for every class, every of the 6 intensities in the moving image, I have one corresponding distribution shown on the top right. And I draw a sample from the distribution. And that's kind of my model. And if you see what sort of forward model you have, that's kind of a sample of the model. It's not very realistic, but it's also not super bad. And so the way this mutual information really works, or one way to look at it is that you actually on purpose, if you on purpose give the wrong deformation field, and you try to predict this image, this is what you get, right? So it's predicting stuff in the wrong place, and there's no structure anymore. And that's kind of the fact that this is bad prediction is why mutual information really works. Or at least that's one way to interpret it. So, okay, let's move away from nearest neighbors. So one trick we did, which actually has some very interesting properties, is that instead of saying, I land somewhere and I need to pick a voxel that is responsible for generating my intensity, I'm just going to select the nearest neighbor. I'm just going to put a little cubic B-spline, and now I'm going to compute probabilities of selecting one of my neighbors. And so the one that's closest to me is going to have the highest probability, but it's not one. I could also pick some that are a bit further away. And there's 4 different, at maximum 4 different ones that you can pick for every pixel. And so if you look at the math, you can actually integrate out over these sort of voxel assignments. Voxel assigning meaning that which voxel has generated, is responsible for generating my intensity. And it turns out what you really do is you take this moving image, which I said has 6 classes, you pull it apart as 6 one-hot encodings, and then you smooth a little bit around the bounds of borders, just a little bit, so you can see it if you zoom in. But the real trick is that now you can use these voxel assignments as latent variables. And we know that cubic B-spline starts to look pretty much like a Gaussian. If you substitute a Gaussian, now you have a quadratic function, which you can optimize with EM. So there we have it. So we implemented it in the GPU. You have non-parametric registration of any type of image without any training at all. There's only one hyperparameter, that's the stiffness of the prior. So we forgot to mention, so we penalize curvature. So it's some sort of smoothing prior, which is Gaussian as well. So essentially on the left, I can maybe quickly explain, if I still have some time, a few minutes. So on the left is essentially the result in the E step, basically. So you say, I have a voxel currently, because of the current deformation field, I land here. And there's a little box of 4x4x4, 64 voxels that could have potentially generated me. And I just compute the posterior probability of each one, and I average their positions. That gives me a deformation field that's unregularized. Then you smooth, and then you rinse and repeat. It's a bit like the Demons algorithm, but from very different principles. And does it work? Yes, it works. So this ongoing work that we're trying to soon submit. So we sort of took data from very different sort of setups. And the experiment we did is one is where you essentially have validation set with segmentation. So you can actually optimize tune parameters based on some validation sets. For instance, if you have CT to MR of the abdomen or of the lung, then you can optimize what are the tunings to actually get the best performance out of this thing. But then we also said, well, what if you don't have any segmentations? And we simulated by just saying, well, you have these tunings for all the other methods. Now you need to segment, let's say, register T1 with a T2 brain scan. Let's just pick randomly one of the tunings that we had on shelf that worked for other cases, and then see how well it works. And then it turns out that actually without any tuning at all, you take this algorithm, no pre-training, no nothing, no tuning, you get 97% in the brain of the maximum achievable performance. So we're pretty excited about that. We're not, obviously we would like to have 100%, no tuning at all, but 97 is not too bad. And then one of the other nice things is that you essentially get a hip sampler for free. So instead of just looking at the mode of the distribution, say what is the single best deformation field that we can find that explains these 2 images, you know, everything works out. You essentially get conditional on, for instance, the voxel assignments. You have that the deformation field is Gaussian, and you can easily sample from that. You go to Fourier domain, add a little bit of noise to the coefficients, and then go back to the spatial domain. And that's interesting because you can actually increase accuracy in different cases by just sampling and using the mean deformation field. And you get automatic error bars, which I try to show in the colors in the middle image there in different directions. What is the uncertainty of the registers? And we're pretty excited about this because this has been a problem that I think nobody has been able to solve so far. So far, most of the uncertainty methods for registration, at least the classical algorithms, is solve sum of square differences. You're using very low dimensional deformation fields because you need to invert some covariance matrix, etc. We don't have that problem. Okay, let me just conclude. So I hope I've convinced you that generative models have some capability to sidestep some of the core problems that we choose discriminate methods. And the second thing is that I've shown you real samples sometimes what the generative models really generate. If you run a forward model, they don't look very pretty. We're not going to win any of the benchmarks that people are organizing about how photorealistic images you generate. But that's really not the case. Not our target. Try to do something other than generating pretty images. Yeah, and maybe this, I don't know what the last thing is supposed to be, but if you're wondering, so each of the process is a researcher and it's an optimization landscape. I think we should, as a community, be a Markov chain in the Carlos sampler, where if the Metropolis Hastings algorithm, if you find something that's a better solution than what you have, of course you go there. But if you find something that's a bit worse, you still should sometimes go there as well. This is just the universal remark. Thanks for your attention.